In this video, I'm going to show you how to use my cellular respiration accounting model. I built this model to help my students understand how cellular respiration works and the flow of matter and energy through a eukaryotic cell. This is the basic interface. These are sliders that you can manipulate, and I have a button down here that you can also manipulate. The target system for this is pretend like there's some probes put inside a eukaryotic cell. And in this eukaryotic cell, there are the enzymes of glycolysis that's located in the cytosol, which then shuttles in pyruvic acid or pyruvate into this complex, the pyruvate dehydrogenase complex, which makes acetyl-CoA. And then acetyl-CoA, which will produce two of those from the two pyruvates, goes through the citric acid cycle, which generates the carbon dioxide. Then you have the electron transport chain along this inner mitochondrial membrane, and that's where most of your ATP is going to be made on this ATP synthase. So let's go back to the actual model and run some simulations of this. So I'm going to click the live button. It's going to give me a live output of data. So this is the initial conditions. I like students to figure out when is glycolysis taking place, when is that pyruvate oxidation taking place, when is the Krebs cycle taking place, and then when is the oxidative phosphorylation along ATP synthase taking place. So let's look at this model, and if you click on it, you can see at the very beginning there is 1 mole of glucose. So what's happening to that is the simulation runs, and then we produce 2 ATP and 2 NADHs. So that's glycolysis. Next, we produce 2 more NADHs but no ATP, but then we generate 2 carbon dioxides. That's pyruvate oxidation. Then we produce 6 more NADHs, 2 more ATPs, and 4 more carbon dioxides, and that's 4 more moles of carbon dioxide. That is the Krebs cycle, or the citric acid cycle. Then all that NADH, along with FADH2, which isn't included here, is used passing electrons to the electron transport chain, which then is used to pump hydrogen ions out of the matrix of the mitochondria. A ton of ATP is produced. Oxygen levels, relative oxygen levels, go down as oxygen serves as an electron acceptor. So that's the basis of the model. It runs basically 1 cycle of cellular respiration during those 20 milliseconds. So let's go through each of these sliders and see what happens. Let's start off with relative oxygen. So when the relative oxygen goes down, you can see the oxygen levels going down, but then at some point, when the oxygen levels are really low, you still get ATP produced, but as a result, it's just through glycolysis. So what happens is your electron transport chain shuts down as there's not oxygen available for to be an electron acceptor, and so this is showing basically fermentation. The NADH gets converted back into NAD during the fermentation process. Take a look at this auto scale here. Your y-axis will auto scale these different quantities, so you have to be careful as you adjust this, the scale auto scale, so make sure you have students be aware of that because it can be misleading. Let's go on to pH. So when I adjust the pH, increasing it, you can see really nothing ends up happening over time. Decrease pH, something else happens, and let's take a look at pH of 5. You can see that the amount of NADH and oxygen really aren't changing, and the amount of carbon dioxides aren't changing, but look at the ATP. It starts skyrocketing at a pH of 5. Here's the thing. Is the ATP synthase at an optimal level at a pH of 5 and then denaturing as we move away from that quantity, or is there something else happening? If you look at the other enzymes, well, they're relatively unaffected by pH changes. So it would be odd that one enzyme would be affected by a pH change and the others not. What can we do to manipulate this to make it interesting is put glucose at 0. So when glucose is at 0, you're still making a ton of ATP. Now this students find very perplexing. Let's adjust the pH back to a 7, and you can see at a pH of 7, nothing's happening. Increase glucose, and back to normal conditions. Decrease glucose, nothing happens. But then when we decrease the pH, ATP starts getting made. Why is that? Well, what happens in ATP synthase? In ATP synthase, hydrogen ions diffuse down their concentration gradient, and that's what's used for oxidative phosphorylation. When I decrease the pH, more hydrogen ions flow as there's a different concentration gradient, and more ATP is made. Let's decrease the glucose again, and you can see how that ATP is generated as there's a greater concentration gradient, more hydrogen ions outside of the space versus inside. Okay, now let's look at this slider, initial NADH. When I increase NADH, you can see the total stock of NADH at the beginning has increased. Well, what does NADH do? Is NADH, it passes electrons to the electron transport chain, which is used to pump hydrogen ions out so that they can flow through to make ATP. So when I increase the original stock of NADH, there's more of an opportunity to make even more ATP through ATP synthase. You still are making the same ATP in glycolysis and the Krebs cycle. By the passing of electrons, as the NADHs go down, they're passing electrons to the electron transport chain, pumping hydrogen ions out, which then makes more ATP. When we reduce glucose to 0, no more glycolysis, no more Krebs cycle, but you still get NADH passing electrons to make a bunch of ATP. Temperature is one of these factors that I included as just part of the causal background. So it's not affecting one system more than another system, but just affecting the overall background of the system. As I increase temperature, it really is decreasing all of these variables. And as I decrease temperature, I'm really decreasing all of these variables. And my idea was denaturing all of these proteins. So with the initial amount of ATP, we can bump this up. And we can see some things interesting happening. And we're just going to run at initial conditions. So this is ATP at 0. And then I'm going to go to this ATP. This is going to show comparative runs. So I'm going to increase this to 20. And as I increase this to 20 and run, so we started off with 20, then 2 are made, then 2 more are made, so that's glycolysis, Krebs cycle, and then more are made during the electron transport chain. We increase to 40 and run. Again, start at 40, 2 are made, 2 more are made, and then we have more made in the electron transport chain. Why I included the ATP is to show something that students often don't have a lot of experience with seeing data with, which is the feedback loop that takes place. So if we look at this system, this reaction right here, where fructose 6-phosphate is made into glyceraldehyde 3-phosphate, is done by an enzyme called PFK, or phosphofructokinase. And this PFK enzyme, when ATP binds to it at high concentrations, it actually inhibits the enzyme. So it has this feedback inhibition where more ATP that's generated, it really shuts down this biochemical pathway. When we increase the amount of ATP, less ATP ends up getting made, and it's not getting made during glycolysis. Look at this as an opportunity to use data to refine students' mental models. Thanks.